Hello everyone, I am Samiksha and today I am going to speak about the GC content in microbial taxonomy. Starting with what is taxonomy? So taxonomy is an area of biological science which comprises of classification, nomenclature and identification. Classification is the grouping of organisms based on the particular characters. Nomenclature is the naming of microorganisms according to the established rules and guidelines by which the organisms are universally recognized. As we know that DNA is the most important tool used in microbial taxonomy, and DNA-based classification is one of the classical genotypic methods. As we know that DNA has 4 bases, 2 purines and 2 pyrimidines. The AT pair is bound by 2 hydrogen bonds while the GC pair is bound by 3 hydrogen bonds. DNA with high GC content is more stable than DNA with low GC content. In molecular biology, DNA-based composition or the GC content is the percent of nitrogenous bases of DNA molecule. This may refer to a specific fragment of DNA or that of whole genome. When it refers to a fragment, it may denote the GC content of a part of gene or group of genes. The formula for calculating the GC percentage is G plus C divided by A plus T plus G plus C into 100, and that for the calculating the AT-GC ratio we can use this formula. There is one more way by which we can easily calculate the GC percentage is this. Using this oligo calculator, if I type any random sequence and I will just have to click on the calculate, then we can get the length of the sequence, the GC percentage and also the melting temperature. Now what is melting temperature? I will talk about it a little later, but I just wanted to show that this is the easy way to calculate GC percent. Moving forward toward the method, that this method can only tell us that organisms are not related. If two organisms have different DNA based compositions, they are not related. However, organisms with identical base compositions are not necessarily related because the nucleotide sequence in two organisms could be completely different. In the given image, we can see that the effect of rising temperature on DNA. Here we can see there is double stranded DNA. When the DNA is heated, the hydrogen bonds start to break. Now it is obvious that the AT bonds will break first and the GC bonds will break a little later. The AT bonds break at around 70 degrees Celsius while the GC bonds break at around 90 degrees Celsius. When the DNA solution is cooled, renaturation may occur and the DNA can come to its original form. Moving towards the methods, I have mentioned two methods to calculate the GC percentage. First one is heat denaturation by using UV spectrophotometry and second is by using HPLC. Now the heat denaturation method is the simplest method. So what we basically do is we heat the DNA sample solution at a high temperature. Then we perform the UV spectrophotometry. So this is the diagrammatic representation. So here we can see here is a source of light, UV light, which can be a bulb or a UV tube. Next we can see there is a monochromator from which the light will pass and the light will disperse and it will move outside this exit slit and pass through the sample. Now here the absorbance is measured at 260 nanometers because we know that 260 range is optimum for DNA as it absorbs most of the UV light. The absorbance is recorded in the detector which is attached to a computer. The next step is to plot a graph. So the graph we can see here, in this graph on x-axis there is temperature in degrees Celsius, and on y-axis there is absorbance at 260 nanometers. DNA solution when heated will indicate a little denaturation, which is approximately below 70 degrees Celsius, but further increase in temperature results in a marked increase in the extent of denaturation. Eventually, a temperature is reached at which the DNA sample is totally denatured. The temperature at which the DNA sample is 50% melted is termed as the melting temperature, or TM. Now what we actually do is we plot the points, we draw the curve, and exactly from the midpoint we draw a straight line. So where the straight line ends, that is our melting temperature, and we just substitute the value in this given formula and we get the GC%. The next method is to calculate GC by HPLC. First, the DNA sample is hydrolyzed. As we know that the mobile phase in HPLC is liquid and the stationary phase can be solid or liquid. As we can see here is a solvent chamber which will have an aqueous solution or any other organic solution. The next is the HPLC pump, which is basically a pressure pump which will move the solution. Here is the injector where we will inject our sample and it will get mixed with the solution present in the solvent chamber. Then it will move in this HPLC column, which will be the stationary phase. It will consist of silica beads. Now our stationary phase is polar and our sample is non-polar, so the sample will get very easily separated and eventually be detected in the detector, and we will get the data in the computer. So this method is very reliable, fast, and can also give us very accurate results. So here is the table where we can see there are different percent of GC content which are highly variable for different organisms. In bacteria, GC percent ranges from as low as 20 to as high as 80%. For example, actinobacteria are characterized as high GC content bacteria, while a species with extremely low GC content is Plasmodium falciparum, which has a GC content of 20%. Organisms that are related have DNA-based composition which is similar or identical. GC content of two organisms, if differ by a small percent, they cannot be closely related. However, similarity does not necessarily mean that the organism is related, since many arrangements of the bases are possible and the genome size and the actual nucleotide sequences also differ greatly. Advantage of using this method is we can easily perform both the methods, but the heat denaturation method is not much reliable. Disadvantage is we cannot specifically identify the exact organism. This method can only tell us that organisms are not related. We can classify organisms into groups, but we cannot exactly identify the organism. For that we will need other genotypic methods like sequencing methods, DNA sequencing methods.